Hello. So today we are going to begin chapter 14, and this first video is going to be on section 14.1 in your textbook. And this section just really gives us an introduction to where we're going with all this material. And the main question we're trying to answer in this unit is, how do we go from a gene to a protein, and how are they connected? So there is a lot of information in this section about how they discovered the way that gene expression works and how genes are expressed. But we're not super concerned with these experiments for the AP exam, but they are interesting if you're interested in them. But essentially what we studied was gene expression. And so gene expression is basically the process by which DNA directs the synthesis of proteins. And a lot of times you might see that it's called from gene to polypeptide because, as you remember, proteins are made up of polypeptides. But typically we call this the one gene, one protein hypothesis because originally they discovered it using enzymes, but they discovered obviously not all proteins are enzymes, and there are proteins that aren't enzymes that code for genes. So we ended up with the one gene, one protein hypothesis. And essentially, the way that this works is through two processes known as transcription and translation. And RNA is kind of the connecting bridge between DNA, our genes, and our proteins that get made. So just a couple things to review about RNA. It's very similar to DNA, but it is single-stranded. It also contains ribose sugar instead of deoxyribose, hence the name RNA versus DNA. And then don't forget that the base uracil, which is represented by a U, is in the place of thymine. So typically in DNA, adenine binds with thymine, or the nucleotide bases match up. With RNA, instead of thymine, you get uracil, U. And so there are two stages, like I mentioned, from getting from our DNA to our protein. One is transcription and one is translation. Transcription is essentially the synthesis of RNA using the information found in DNA. The way you can think of this is we have DNA, and that's a nucleic acid, and then we have RNA, also a nucleic acid. And these two nucleic acids are essentially written in different forms of the same language, the nucleotide bases. So when we're making RNA, essentially we are transcribing the language in DNA to RNA, hence the name transcription. And in transcription, DNA serves as the template, and then basically you assemble complementary sequences of RNA. And then the mRNA is the product of transcription, and that mRNA carries the genetic message from the DNA to protein synthesis, known as translation. So translation is the synthesis of a polypeptide, aka a protein, using the information from mRNA. And in this case, we're now going from a gene, or the information in the nucleic acid, the nucleotide bases, to a protein. So this language must be translated, hence the name translation. And then if you recall, ribosomes are where translation actually happens. We have studied a lot about transcription and translation in bacteria and eukaryotes, so we're going to focus on those in this section. There are some slight differences. So one of the biggest differences is that in bacteria, the translation of mRNA can begin before transcription has finished. And that's because, if you remember, in prokaryotes, there's no nucleus. So the DNA is just free floating. And so there's no compartments where different things happen. So in fact, the translation of mRNA in bacteria and other prokaryotes can sometimes happen simultaneously while transcription is still occurring. In eukaryotes, you'll recall they do have a nucleus, and so all the transcription in DNA to RNA pieces happen in the nucleus. And then the resulting pre-mRNA has to be exported to the cytoplasm, and that is where it gets transported, and then translation can begin. And even before that, before they send it out to the cytoplasm, the mRNA in eukaryotes undergoes some additional processing. So we have here a bacterial cell and a eukaryotic cell. So you can see in the bacterial cell, basically you go from DNA to RNA to the protein, transcription to translation, all almost simultaneously or as it's happening. In the eukaryotic cell, so this would be, for example, in an organism like humans, transcription and RNA processing happens in the nucleus to get our mRNA, and then you'll notice translation occurs in the cytoplasm. So that's what's happening here. Then once we get our initial RNA, it's also known as the primary transcript. But really, this whole section is about something that Francis Crick termed the central dogma of gene expression. And that essentially, the central dogma, is that we start with DNA, it is converted into RNA, and then from there it becomes a protein. And you can think of these proteins as the expression of our genes. So how exactly does this work? Well, there is something known as the genetic code which makes this possible. So there are 20 amino acids and only 4 nucleotide bases in DNA. So one question scientists had to answer was how many nucleotides give us one amino acid? And they eventually found that this genetic code is a triplet code. So there is a series of almost, you can think of it like non-overlapping words made up of 3 nucleotides apiece, and each of those forms a triplet. That code is transcribed to a complementary 3 nucleotide code in mRNA. So the DNA has a triplet code strand that serves as the template, and the mRNA makes a complementary section, and then that will then be translated into an amino acid forming a polypeptide. So basically, you have 3 nucleotides coding for 1 amino acid. And this is a little bit about how that might work. So we've got our DNA molecule with all these different genes. We're going to zoom in on gene number 3. Here's our DNA template strand. In the next section, you'll learn about transcription. You get the complementary pieces. So you see U goes with A, C goes with G, and so on. And then each of these 3 nucleotides, that's one of our codes, known as a codon, is the code for an amino acid. And you can see the UGG codes for tryptophan, for example. And we kind of already talked about how this happens. And the template strand is the strand of DNA that provides the template for the RNA, and that's the same no matter the gene. As long as it's the same gene, the template strand is going to be the same, because then you get the expressed proteins. As I mentioned in the picture, the mRNA-based triplets are called codons, and those are read in the 5' to 3' direction. And remember, each codon is the set of 3 nucleotide bases that codes for a specific amino acid, and so that's very important. There are there's 64 codons, but out of those, only 61 code for amino acids. The other 3 are known as the stop codons, and they are what signal the end of translation. So in this case, there's 3 different codons that don't code for any amino acid, and that's the signal to stop. This genetic code is very redundant, so there's a few different codons that code for 1 amino acid, which I'll show you a picture of this in a moment, but it's not ambiguous because every codon only codes for 1 amino acid. And this is really interesting evolutionarily because regardless of the organism, from us to bacteria, the genetic code is nearly universal. So those same triplets code for the same amino acids whether you're a bacteria, a plant, a human, a moose, it doesn't matter. Um, and they have to be read in their non-overlapping fashion. So every set is a set of 3, and that is one codon. The next one will start with the following set of 3. So you can see here, once you go to college, you're probably going to have to memorize the codons for every amino acid, but for the AP test, you don't have to do that. Just notice here, when you see, for example, like uracil, 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 that codes for this amino acid. When you see uracil, uracil, guanine, or uracil, uracil, adenine, that codes for the amino acid leucine. And all of these have different codes. You can see there's different ones for different amino acids, but we have 3 stop codons, and then AUG technically codes for the amino acid methionine, but it can also serve as the start codon, which will come in handy when we talk about transcription. So that's where we're going to stop for the intro, and the next video will be about transcription.